To today's webinar on research for families, biomarkers, bone density, and behavior. Before I introduce today's presenter, I want to make a couple of announcements. We know many of you are listening in from far away and aren't able to attend local events or programs, so that's why we provide this free webinar series. We host several webinars per month, so please visit our website to view the current schedule. Simply visit our website at www.johnson-center.org and click on the webinars link on the right-hand side. The 2018 webinar schedule will be added this week, so if you are not on our email list, I encourage you to visit our website and click on the Join Our Email List link that appears on the homepage. To get instant news and events from the Johnson Center, follow us on Facebook, Instagram, and Twitter, as we often announce grant and scholarship opportunities, research opportunities, and special events and presentations there. And be sure to check out our YouTube channel at youtube.com/thejohnsonCenter. Please note that while we don't send out copies of the slides of the presentations, all past webinars are posted to the YouTube channel and accessible for free. You'll find a library of a wide variety of topics, and links for certificates of attendance are in the webinar description. Be sure to follow our colleagues at the Autism Research Institute as they host their own webinar initiative, and they share some great resources on their website and social media pages. For those of you in and around Austin, be sure to mark your calendars because the Santa and Mrs. Claus will be in town on December 12th. Join us for our holiday open house and enjoy allergen-friendly treats, crafts, story time by Mrs. Claus, and photos with the big guy himself. You'll find details on the blog section of our website and social media pages. If you would like a certificate of attendance after today's webinar, look for a follow-up email in your mailbox 1 hour after the webinar concludes, or look for the link on our YouTube channel in the webinar description. It will contain instructions on how to get your certificate. If you have questions during the live webinar, you may type them into your GoToWebinar control panel. If you are watching a recording of this webinar, you may email questions to the presenter at info@johnson-center.org. Today's webinar will be presented by Dr. Laura Hewitson. Dr. Hewitson is the Director of Research for the Johnson Center for Child Health and Development. She has published over 65 peer-reviewed scientific papers, invited reviews, and book chapters, and she has presented her work at many national and international conferences. Dr. Hewitson oversees a diverse research portfolio for children with autism, focusing on immune dysfunction, nutrition, and the development of biomarkers as both diagnostic tools and as possible therapeutic targets. Please welcome Dr. Hewitson.
Good afternoon, and thank you for joining us today. I will be presenting a webinar on autism research for families, biomarkers, bone density, and behavior. This presentation will be focusing on the clinical research that we are conducting at the Johnson Center. It is primarily geared towards parents, although researchers and clinicians may also find the presentation of interest. Feel free to submit your questions during the presentation using the GoToWebinar website, and if there is time at the end, I will try to answer them. The objectives for this presentation will include a general discussion on biomarkers, how biomarkers can be used in autism research, the challenges we face in developing biomarkers for autism, a summary of our clinical studies, and a section on how you can help advance autism research. Although the term biomarker is relatively new in the field of autism, biomarkers play a major role in medicine and have therefore been used in clinical diagnosis for considerable time. For example, body temperature is a well-known biomarker for fever. Blood pressure is used to indicate the risk of stroke. Cholesterol values are a risk factor for either vascular or coronary disease. And C-reactive protein is an indicator of inflammation. There are several definitions for the term biomarker. This one was developed by an NIH-sponsored working group, and it describes a biomarker as a characteristic that is objectively measured and evaluated as an indicator of normal biologic processes, of pathogenic processes, or pharmacologic responses to a therapeutic intervention. To break this out further, I'm going to highlight a few of the terms used. First of all, characteristic. This essentially describes anything that can be detected or measured. It can be a physiological response, such as body temperature or blood pressure. It could describe compounds in the tissues or body fluids, such as nucleic acids or hormones. It could refer to cells, such as cancer cells or cells of the immune system. And it could refer to measurements of body structure. Perhaps the most important consideration when describing a biomarker is their objective nature. Objective meaning reliable and accurate. This is in contrast to many measures used in clinical practice, which, while often referred to as a biomarker, they are inherently subjective, requiring an evaluator to interpret their meaning. We are in an era of highly innovative imaging and molecular technologies, and it is these technologies that allow us to objectively measure biomarkers. They include technologies such as structural and functional imaging, as well as a number of the omic technologies such as genomics, proteomics, and metabolomics. There are a number of different kinds of biomarkers in medicine that can be evaluated. Biomarkers may be diagnostic, meaning that they can detect the presence, stage, or type of disease. They may be prognostic, by providing information on a clinical outcome in the absence of specific treatment. They can also be predictive, meaning that they may predict a clinical response to a specific intervention. And they can also be pharmacodynamic, providing information that may indicate a relevant response to a treatment or intervention. Finally, some biomarkers are measured following an intervention or treatment. While pharmacologic responses are the ones most commonly measured, the intervention can also be behaviorally based, such as ABA. It can be assistive technologies, such as a device or a service. And it can include a whole range of other interventions, including physical and occupational therapy outcomes and dietary intervention. There are several important applications for a biomarker for autism that are currently being researched. These include biomarkers of ascertaining risk, diagnostic and predictive biomarkers, and biomarkers to identify targets for treatment or to monitor treatment efficacy. Looking at these individually, let's first look at ascertaining risk. Biomarkers can provide a reliable screening tool for infants and children to determine risk. Twin and family studies have demonstrated that both genetic and non-genetic factors contribute to an increased susceptibility to autism. However, despite the clear implications of genetic factors in the etiology of autism, none of the genes or genetic variants that have been so far identified can be considered clinically useful for assessing autism risk with a high degree of certainty in a large percentage of the population. Non-genetic factors that may contribute to autism risk include preconception factors, such as advanced parental age, parental autoimmune disease, season of birth, spacing of pregnancies, birth order, and even fertility treatments. It can also include prenatal factors, such as multiple births, maternal infection, maternal health and diet, prescription drug use, and pregnancy complications. And finally, risk factors can include postnatal factors, including premature birth, low birth weights, complications during childbirth, and many different environmental exposures through early childhood. There are many different avenues to explore in examining risk factors for autism, and this is where a great deal of current biomarker research is focused. Biomarker research is also looking at diagnosis, as biomarkers may be useful for improving the reliability of clinical diagnosis tools. Currently, the diagnosis of autism is defined on the basis of behavioral criteria. However, the condition is associated with a wide range of comorbid conditions and other biological phenomena. It is hoped that by translating markers of these phenomena into clinically useful biomarkers, it will help to improve the validity and efficiency of existing diagnostic methods. Biomarkers may also be useful as a predictive biomarker while a child is pre-symptomatic, meaning that they haven't yet shown any signs of developing autism. The behaviors that are characteristic of autism first emerge and then evolve over the first few years of postnatal life. In many cases, though, it is quite difficult to distinguish autism symptoms from just normal variations in development, or from transient delays that then resolve over time. It is hoped that valid biomarkers that are identified before the onset of clear behavioral symptoms will help in the early detection of emerging autism. And finally, biomarkers may be useful for identifying novel treatments, to confirm the need for a specific treatment, or to monitor treatment efficacy. The majority of studies examining autism biomarkers are focused on ascertaining risk based on maternal or in utero markers. For most of these measures, it is not yet clear if the abnormal biomarker is a contributing factor to the development of autism or just a result of another underlying abnormality. While the precise mechanisms of the association are debated, they likely include defects in oxidation, oxidative stress, inflammation, hypoxia, ischemia, endocrine disruption, neurotransmitter alterations, and interference with signaling pathways. Regardless, the future of autism research could involve a broader view of these biomarkers, which might hold more value in combination than in isolation. As a result of new technological advances, it is now possible to use a machine learning technique that is trained to identify complex patterns of data that can help make predictions. This approach could be generalized to certain groupings of biomarkers, which, where individually insignificant, when analyzed together, they might generate a pattern of clinical relevance like diagnosis, severity staging, or response to treatment. These techniques might also be able to identify the most relevant or most predictive biomarkers among the many biomarker candidates thus far proposed. However, there are many challenges that are impeding the progress in the development of biomarkers for autism. A significant challenge in identifying these biomarkers is that they may reflect complex genetic or biological changes, or even epigenetic processes that are only active during particular periods of time, and they do not actually define the disorder, only the process that led to it. These genetic and epigenetic features contribute to an individual's phenotype. This is a person's observable characteristics, such as their behavior and development. While phenotypic characterization of autism is an integral part of advances in clinical practice and research, a given phenotype may arise from a diverse set of biochemical processes. Therefore, the treatment of a phenotype with a specific drug or intervention might be extremely effective for one individual with a given set of genetic and/or epigenetic biomarkers, but completely ineffective for another with a different pattern of these biomarkers. Defining this complex multidimensional phenotype for autism is very difficult. Currently identifying behavior symptoms through standardized assessments Semi-structured play-based interviews or questionnaires, combined with clinical judgment, are the gold standard for making an autism diagnosis. However, an inherent limitation of this diagnostic method is that a child must be of a certain age in order to assess the absence or presence of critical diagnostic behaviors. There is also a lack of agreed and stable definition of autism. In fact, the changes to the DSM-5 diagnostic criteria for autism suggest that the precise behavioral definition of autism is still in its flux. The clinical phenotype of autism is widely heterogeneous, giving rise to a spectrum disorder. Despite common areas of impairment that may define autism as a condition, individuals with autism show a vast clinical variability in the expression and severity of their symptoms. The general consensus over the last few decades is that this heterogeneity leads to a single spectrum disorder. However, researchers have now hypothesized that this heterogeneity represents many distinct behavioral and physical symptoms, which therefore lead to multiple etiologies rather than a single unifying cause for autism. The heterogeneity of autism encompasses individuals with varying presentation of symptoms that are not fixed throughout the course of life. While every individual on the autism spectrum has problems to some degree with social skills and empathy, communication, or behaviors, the level at which this impacts their functioning and the combination of symptoms each person presents with varies tremendously between individuals and also at different ages. There is also considerable heterogeneity in the associated medical conditions and non-diagnostic symptoms in autism. For example, as many as 25 to 33% of individuals with autism develop epilepsy, and 55 to 70% have an intellectual disability. Individuals with autism may or may not have a sleep disorder, may have eating disorders, intestinal disorders, motor skill deficits, or even tics. Between 24 and 59% of individuals with autism have an anxiety disorder, and 28% meet the criteria for ADHD. These comorbid factors pose further challenges for researchers in biomarker development. There are also a number of other factors that should also be considered in the search for biomarkers. These include age, gender, specificity, and sensitivity. It is highly likely that different biological systems in autism may be at play at different time points of development. In applying this insight to biomarker development, it follows that studies conducted in individuals of different ages may yield distinct results. So to meet this challenge, it will be necessary for researchers to conduct biomarker studies in the context of tightly developmentally constrained research samples, or to analyze biomarkers with samples sufficiently large to permit analysis of developmental effects. However, both of these approaches are quite challenging. A strong male bias in autism prevalence has also been observed with striking consistency, but no mechanism has yet to definitively account for the sex difference. Some biomarker studies therefore tend to enroll more males than females, or focus only on males. However, there are a few studies that have identified biological markers that are differentially expressed in males and females with autism, suggesting that we may need to develop gender-specific biomarkers. However, based on this gender bias in autism prevalence, it takes much longer to enroll girls with autism for the research studies. Since autism is a very heterogeneous disorder with wide variation both in its core diagnostic features and also in associated characteristics, the search for biomarkers of autism per se may be unsuccessful for the majority of etiologies. For example, Repetitive and restricted behaviors, a common feature of autism, are also a feature of obsessive-compulsive disorder. Similarly, disruptive social cognition, also a common feature in autism, is evident in schizophrenia and related disorders. Furthermore, the results of autism research studies most commonly reflect differences in means between groups of individuals, your controls and your affected individuals. In this way, a difference between groups typically reflects a shift in the distribution of values in a biomarker parameter. However, when results are reported in this fashion, it provides little information about the relevance of a biomarker at the individual level. So unless a biomarker has high specificity, meaning it is true for all people with autism and is specific to people just with autism and no other diagnoses, analyses of this nature provide little practical information. When examining autism biomarkers, a high sensitivity is also required. This means that autism will be identified in most of the individuals being studied. Since autism is considered a spectrum disorder, those individuals with very mild symptoms, but still meeting the diagnostic criteria for autism, may not be positive for a potential biomarker that is found in people with some more severe symptoms. Both still have an ASD diagnosis, but the biomarker in question may not be sensitive enough to identify ASD in all subjects. So I've presented a lot of information on some of the scientific challenges that we are faced with in biomarker development for autism. At the very minimum, research requirements for developing these autism biomarkers need therefore to include rigorous diagnostic assessments with extensive phenotyping, subgrouping for comorbid conditions, age and gender, and also the inclusion of appropriate controls. There have been a number of proposed biomarkers for autism. This isn't an exhaustive list, but it includes head size, brain structure and function, eye tracking, vocal acoustics, and a wide range of biological candidates. This represents just some of the proposed biological markers or targets for autism. This is studying single metabolites now as potential markers. Although study outcomes are not always consistent, and this is because inclusion-exclusion criteria are often different across studies. The diagnosis is not always confirmed using gold standard methods, and sometimes controls are not even included in the studies. This has led to a long list of putative biomarker candidates, some with very little clinical utility. It appears that immune dysregulation and biomarkers associated with this appear to be one of the most consistent mechanisms proposed as generating potential biomarkers. This is based on research examining immunoglobulins, cytokines, autoantibodies, and inflammatory markers in children with autism. Certainly a biological marker that could predict ASD risk, assist in early diagnosis, or even identify potential therapeutic targets would have great clinical utility, especially since current diagnostic methods and screening tools are somewhat subjective, and they are quite difficult to assess in younger children, which can often result in missed opportunities for early intervention. And this also assumes that each person has access to these diagnostic methods, whereas we know from research that not everybody has access to doctors that can provide this diagnosis. I'm now going to describe some of our clinical research studies, particularly those involved in biomarker development. Biomarker studies have primarily been focused on proteomic approaches, looking at nutritional imbalances, and also looking at their association with behavioral domains in autism. The proteomics approach that we have used is based on multiplex assays to screen blood samples from children with and without autism for potential proteins that may be specific to autism. These types of assays can simultaneously measure hundreds or even thousands of analytes in a small sample of blood. This allows us to cast a wide net so we can identify as many possible biomarkers. Once we have identified these biomarkers, they then need to be validated. This typically means you then examine new blood samples to see if the biomarkers you have identified can further distinguish blood samples from children with and without autism. For this study, we used a company called Myriad Rules-Based Medicine, which is based here in Austin. They have developed several disease-specific multi-analyte profiling tools for biomarker discovery. This work was recently published in the Journal of Neuroinflammation and can be found online. In this study, we used serum samples from boys ages 2 to 8 years of age, both with and without autism. We chose only to use samples from boys since we had a much larger number of boys enrolling in our studies. There is also mounting evidence that autism in boys may be different than autism in girls in several ways, particularly in biological markers. Any subject diagnosed with either a genetic or metabolic condition or other concurrent physical, mental, or neurological disorders were excluded, as were subjects that were currently taking psychiatric medications or had taken psychiatric medications within the last 3 months prior to enrollment. All subjects were considered relatively healthy with no reported illnesses for 3 weeks prior to participation in the study. For the ASD group, the subjects were assessed by a psychologist trained in research reliability using both the Autism Diagnostic Observation Schedule, the ADOS, and also the Autism Diagnostic Interview, the revised version. Both of these are considered gold standard for ascertaining an autism diagnosis. This diagnosis is then made based on all of these data and also the overall clinical impression by the psychologist. These children were diagnosed using DSM-4 criteria, as when we first began enrolling subjects on the study, the DSM-5 diagnostic criteria had only just been approved, and so for this particular study, the majority of our subjects had already had a DSM-4 diagnosis. We also excluded those subjects with a diagnosis of Asperger's syndrome or pervasive developmental disorder not otherwise specified, PDD-NOS. So only boys with a diagnosis of autistic disorder were included to try to improve the subgrouping and also decrease our heterogeneity. For the control group, all subjects underwent a developmental screening using the Adaptive Behavior Assessment System that was also assessed by the psychologist. Control subjects were excluded if their score on the ABAS suggested possible abnormal development and the need for further evaluation. Controls were also excluded if they had a first or second degree relative diagnosed with autism. This is done to try to improve our chances of identifying autism-specific markers and removing that familial aspect. So we looked at 184 analytes using the rules-based medicine platform. 11 serum proteins were found to be differentially expressed in blood samples from children with and without autism. 6 of these analytes were found to be significantly different between blood samples from children with and without autism at a significance level of less than or equal to 0.05. Thyroid-stimulating hormone and interleukin-8 showed the greatest % change between ASD and control samples within our p-value. We therefore focused our current studies on those 2 analytes. So let's learn a little bit about TSH and IL-8. Thyroid-stimulating hormone is a pituitary hormone that stimulates the thyroid gland to produce thyroxine, and then triiodothyronine, which stimulates the metabolism of almost every tissue in the body, so it is very, very important process. TSH is secreted throughout life, but it reaches high levels during periods of rapid growth and development. Thyroid hormones are essential for brain maturation and for brain function throughout life. In fact, thyroid hormone deficiency, even for short periods, can lead to irreversible brain damage, the consequences of which depend on the specific timing of the onset and also the duration of thyroid hormone deficiency. Reduced levels of thyroid-stimulating hormone have also been reported in studies looking at blood spots from infants who are then later found to have autism, providing additional evidence that TSH levels may be abnormal early on in development and could therefore be a useful indicator as an early biomarker for autism. More recently, studies have shown maternal mid-pregnancy serum levels of TSH are inversely correlated with the likelihood of having a child with autism, meaning lower TSH levels during pregnancy are associated with higher risk of having a child diagnosed with autism. Several studies looking in children have also looked at TSH levels, but the results were not always consistent. However, typically children with autism do tend to have lower TSH levels than control children, as our study would suggest. The protein levels for TSH were regressed on each of the ADOS subdomain scores and also with phenotypic subgrouping to examine whether the levels of TSH reported were related to a clinical measure of autism or related comorbidities. We found that while TSH levels were not correlated with phenotypic subgroups, such as GI concerns, verbal ability, and the type of ASD onset, more severe scores on the ADOS in both the communication and social interaction domain and also the stereotype behavior and restricted interest domain were correlated with lower levels of TSH. This suggests that TSH may be a useful biomarker in behavioral index of ASD in boys. The second biomarker that we looked at that showed differential expression between our groups was interleukin-8, or IL-8. This is a pro-inflammatory cytokine, which is a type of signaling molecule that is excreted from immune cells like helper T cells and macrophages. Pro-inflammatory cytokines are predominantly produced by and involved in the upregulation of inflammatory responses, so they have been directly linked with neuroinflammation. A number of pro-inflammatory cytokines have been reported to be elevated in children with autism, including IL-8, although the vast majority of studies identify both IL-6 and IL-1 beta to be those consistently elevated across studies. There is less information known about IL-8, although elevations in plasma IL-8, remember this is a serum study we performed, have been reported in some high-functioning boys with autism. Elevations in IL-8 have also been reported in blood spots collected at birth in children that go on to develop autism, suggesting that these IL-8 levels may also be useful as a very early biomarker for autism. We then went on to validate TSH and IL-8 levels on using a different platform, MESOSCALES delivery. This provides greater sensitivity than the rules-based medicine Luminex platform we used earlier. This again showed us that TSH was about 30% lower in children with autism, and IL-8 was 16% higher in children with autism, in boys with autism, which was consistent with our first findings. We then looked at the predictive accuracy of identifying a blood sample from a child with autism using just TSH alone or using IL-8 alone, and they were about 75%. However, when we pooled these two proteins together and combined them as a panel, the predictive accuracy is identifying a blood sample from a boy with autism went up to 82%. It was quite interesting that the two serum biomarkers that had the most significant change between ASD and control children in our study have both been shown to be altered in newborns, suggesting that in the future we may be able to include an autism risk screen in that newborn blood spot screen that is routinely collected at birth. This provides more evidence that at least in a subset of children with autism, a type of inflammatory response begins during pregnancy and is likely a trigger for autism. This trigger gives rise to an emerging model of neuroinflammation. Although this slide is a little complicated, I'm going to talk you through it, starting at the top left with maternal infection. Maternal infection during pregnancy can cause a number of different responses. Some pathophysiological responses, such as the production of immune factors like cytokines, reactive oxygen species, and also soluble endocrine factors such as stress hormones, have been detected. Some of these factors then can cross the placental barrier and enter the fetal environment, thereby causing fetal inflammation and oxidative stress. Another mechanism might include maternal infection causing temporary states of macronutrient and micronutrient deficiency. These can limit the fetal availability of essential nutrients necessary for normal fetal development and growth. And finally, a third manner in which maternal infection can impact the fetus is the microbial composition of the placenta. We've heard a lot about the microbiome and autism over recent years. Well, it is possible that maternal infection during pregnancy can have effects on the placental microbiome. This would then alter the development of the offspring's microbiome and thus predispose the developing child to dysbiosis and other microbiome-associated abnormalities. This model therefore provides plausible evidence that neuroinflammation begins in utero and may be a trigger for autism. Further evidence of this neuroinflammation has been shown in studies of older children diagnosed with autism, where inflammatory signals dominate, anti-inflammatory responses are inadequate, and a chronic activation prevails. The more skewed towards inflammation, the more acute the autism symptomology. This dysregulation has also been demonstrated in brain tissue from children with autism, where the cells that help maintain the neurons, these are called astroglia and microglia, are enlarged from a state of chronic activation. Pro-inflammatory signaling molecules are then increased, and genes involved in inflammation are switched on. These findings are important for many reasons, but perhaps the most noteworthy for biomarker research is that they provide evidence of an abnormal, continuing biological process, which means that there is finally a therapeutic target or multiple targets for a disorder that is typically defined by behavioral criteria. So what are the next steps for this study? Well, we have already shown that the success in identifying blood samples from children with and without autism can yield different biomarkers, and it will go up if we include both TSH and IL-8 in our panel. So one approach would therefore be to continue looking at some of the additional proteins that were first identified, such as ApoE and CGA, both of which have been implicated individually in autism. Increasing the size of a biomarker panel may therefore increase both the specificity and the sensitivity, both of which are important for biomarkers. We are also looking at running blood samples on US multiplex platforms that can screen thousands of proteins. This may provide novel biomarkers that we haven't yet screened. And then also we would very much like to screen blood samples from both girls and boys, which will be important for identifying any gender-specific biomarkers, and also including sibling controls as well as non-sibling controls, as this will help separate out familial markers from autism risk biomarkers. So switching gears a little now, I'd like to talk about a recent study that we have completed in looking at bone density in children with autism. We know that low bone mineral density in children with autism has been reported in several studies. However, these authors speculated that this was probably caused by nutrient deficiencies due to dietary restrictions, such as a gluten and dairy-free diet. However, we know that children with autism are at greater risk of GI disorders than their typically developing peers. And so it could be these GI concerns that may be impacting their nutrient availability. While there is limited data on the impact of GI symptoms on bone health in children with autism, decreased bone mineral density in children with inflammatory bowel disease but no developmental concerns is well established. So severe GI symptoms in children with autism could compromise dietary intake and cause nutritional depletion. This in turn might negatively impact their growth velocity and therefore increase the risk for suboptimal bone development. So in this study, we enrolled boys that were 4 to 8 years of age, and we performed a DEXA scan of the lumbar areas of the spine to evaluate bone mineral density, as well as examining a number of nutritional factors such as vitamin D levels, calcium levels, whether the child was on a restricted diet, such as a gluten-free and dairy-free diet, and whether there were GI concerns in the children. The results from this study were recently published in the Journal of Autism and Developmental Disorders. I'm going to briefly summarize the findings for you. Bone mineral density was lower in boys with autism, but contrary to other studies that could only speculate about risk factors, this study showed that low bone mineral density was not correlated with the dietary intake of calcium and vitamin D, or with elimination diet status, meaning that children that were on a supported GFCF diet were not at greater risk for lower bone mineral density. Although low bone mineral density was not correlated with GI symptomology, children with autism on this study reported significantly greater GI concerns across all of the domains assessed compared to children without autism. Kelly Barnhill, the PI of this study, will be discussing this data in much more detail in a webinar in the new year. You can either check out our 2018 webinar schedule, which will be posted within a week on our website, or you can sign up on the Johnson Center homepage to receive emails, which will include notifications about our webinar schedule. Unfortunately, I don't have time to go into more details on some of our other studies that have been published, but this slide represents some of the more recent clinical studies. Many of these are available for free online. If you put my name into the search bar on PubMed, you can generate a similar list to this and then download the studies. I'd like to end this webinar discussing how you can help advance the status of autism research. If you have not done so already, please consider participating in a research study. Information on available studies that are currently enrolling can be found online at multiple different university sites and nonprofit sites, through different community groups, and through autismclinicaltrials.net. One of the biggest hurdles facing researchers in performing research studies is the enrollment of study participants. This is true for all areas of clinical research, such as AIDS, heart disease, type 1 diabetes, and of course autism. Many families are just not interested in having their child or even themselves participate in a research study. But study participation is at the heart of all medical advances. Clinical research offers the hope for families and an opportunity to help researchers find better treatments for others in the future. In a recent CS-Mart Children's Hospital national poll, they reported that 77% of parents want only FDA-approved medicines for their children, and that they value the information that comes from research. However, only 30% of these parents said they would be willing to have their children participate in medical research. This discrepancy has to change in order to improve research study participation. I would encourage you to talk to your kids about volunteering. Don't underestimate them, as many kids really enjoy the opportunity to help other kids. If your child is anxious, we have a social story available that can help them with preparing for an office visit or even a blood draw. For those of you who live locally, we have an easy research opportunity for your family to participate in. Our nutrition study is examining whether dietary status differs between children with and without autism. We are currently enrolling boys and girls between the ages of 18 months and 8 years of age for this study. Once consent has been reviewed and given by the parents, they are provided with questionnaires to fill out that ask questions about their child's health, development, and the foods they eat. A short office visit is required, where we will measure the child's height, weight, and also arm muscle measurements. It takes about 10 minutes, and that is all that is required. Compensation is provided, and all children receive this cute superhero t-shirt. In fact, I know of one mom who shared with me that their child wore their superhero t-shirt for 2 weeks solid. Our nutrition study is a great way to introduce your child to the process of participating in a research study in a relaxed and stress-free environment. After completing in the nutrition study, we usually find children are much more willing to participate in our biomarker study. They've had a good experience participating in research, and they're willing to try the biomarker study. We are enrolling children, both boys and girls, that are 2 to 8 years of age. We're enrolling children both with and without autism, so that will include healthy children that do not have autism. After reviewing and signing the consent forms, several questionnaires are provided to the parents to complete. However, if you've already participated in the nutrition study, you do not need to complete these again. For children with autism, diagnostic assessments are performed by our clinical psychologist if these have not been performed within the past 12 months, in order for us to verify or confirm a diagnosis. During an office visit, we measure height and weight, and then the child undergoes a blood draw. This is performed by one of our highly trained and very gentle nurses who have a lot of experience with children both with and without autism. We even have a TV where kids can watch their favorite movies during the procedure. Compensation is provided, and all children receive the superhero t-shirt. If you have already participated in one of our research studies or in other autism research studies around the country, thank you. You may be asking, what else can I do? Well, in the CS Mott Children's Hospital poll I mentioned earlier, 92% of parents said that they had never been asked to have their child participate in a research study. So please consider sharing your story. If a friend or a family member asks how they can help you, ask them to consider having their child participate in an autism study, or better yet, provide them with one of our flyers. If your son or daughter has participated in a research study, have them explain to their friends what it was like. For more information on our current research studies, please check out our website at the URL listed. Additional information on our past webinars can also be found online. You can check out our YouTube channel or look for the Johnson Center 2018 webinar schedule, which will be available shortly. I'd like to thank you very much for your time and attention. I'm going to spend just a couple of minutes now looking over the questions that have been submitted during this presentation, and then I'll come back to you to hopefully answer some of those. Okay, first question I'm going to answer. Do you have any ongoing studies for older children with autism? We do, but there are some pretty strict inclusion-exclusion criteria to participate in those studies. We have one study that is based in the Rio Grande area of Texas. This is examining nutrient status again, but it's in children from 18 months all the way up to 21 years of age. And it's for children that have any type of developmental disability, a developmental disorder, but also those with complex medical diagnoses. It's in collaboration with one of our partners, Milestones Therapeutics, and you do have to be a patient of Milestones in order to participate. So if you are in the Rio Grande area, down in the very south part of Texas, and have access to Milestones Therapeutics as a therapy center, then please contact them and ask them about the study. You can also check back on our website periodically, because we do sometimes have studies that go up to 18 years of age or even 21 years of age. Another question: based on all of the information you provided, it seems unlikely that a biomarker for autism will ever be identified. Can you please comment on this? Yes, I hope I haven't presented too much doom and gloom for biomarkers for autism. That was not my intent. It is certainly true that it's going to be very challenging to identify a single blood biomarker that would perhaps be able to predict autism with a very high specificity and sensitivity, say over 90–95%. However, I do think as we gain more information from all of these studies that are being published on metabolic and immunologic alterations, especially during the prenatal period, identifying a biomarker, or more likely a panel of biomarkers that could indicate a high risk of autism, would probably be very useful in a newborn screening tool. I mean, we already have a number of biomarkers that we know are altered during pregnancy. So these could be part of that newborn screening tool, or even those that are being identified in children at a later age could be potentially put into a newborn screening tool. However, we do need more studies that will be examining blood spots in children, and then we have to track those children for several years to see which children go on to develop autism and those that don't. So you can see that these studies will take a lot of time to complete, but I think this is perhaps the most likely approach that will be successful for developing an autism biomarker. It'll be part of a screening tool, perhaps, for newborns. Oh, this is a good question. I didn't address this in the study. Can my son participate in the biomarker study? We live in California. I wish the answer to that was yes, as this would certainly aid in our subject recruitment. But at this time, we can only enroll subjects that are based here locally and through our center. This is the reason for this is because blood samples that we include in our studies are processed under very strictly controlled conditions. That includes the time from collection to being handed over to our research assistant who begins the processing of the samples, the timing and the speed of the actual centrifugation. That can vary dramatically from lab to lab, and we want to keep that very consistent. Also the temperature of the sample during the processing. And then the storage of the samples, we get them into -80 freezer very quickly, and that is not typically available in many research labs, in many clinical labs. So unfortunately, commercial blood draw facilities like Quest and CPL, I know those are here locally, or whatever you have in California, they don't have the time to do this, or perhaps even the inclination to do this and process samples under the strict requirements that we need for research. If there are any clinicians or researchers listening in today and they have access to a large number of patients that they're collecting blood samples routinely, we'd love to talk to you. Perhaps we could set up a second center in a different location that could also collect samples, and we could collaborate on this research together. It's now just a few minutes before 1, so I think I'm going to end this. But again, I'd like to thank you very much for your time and attention. If I haven't addressed your question, you can send it to johnson-center.org, and I will try to address those individually. And thank you very much for listening.